So what we're going to try to do today is we're going to try to pick up on where we left off last time and extend the ideas of generating a protein that's going to end up free in the cytoplasm. That is, those proteins that we talked about last time were those that were produced on free ribosomes. Remember, we divided early on our proteins into those proteins destined to go into the cytoplasm and those destined to go into the endoplasmic reticulum. The endoplasmic reticulum proteins were going to be the lysosomal proteins, secreted proteins, and transmembrane proteins. And the rest of them were going to be made on free ribosomes and put into the cytoplasm. So now the question is, what are the mechanisms that sort these two protein types? And that's what we're going to try to explain today. And we're going to start here with the idea of targeting proteins to the endoplasmic reticulum. There are a couple of fairly straightforward rules for this. The first is that the generation of a signal peptide, usually the first 20 or so amino acids of the protein, if these proteins are hydrophobic, they generate a signal peptide. The signal peptide, because it's hydrophobic, because of these hydrophobic sequences of amino acids, these then can bind to another protein that's in the cytoplasm. This is called a signal recognition particle, the SRP. The SRP binds to the signal peptide. So what we're going to see is hydrophobic interaction between the signal recognition particle and the hydrophobic amino acids on the protein that's being generated as it comes out of the large ribosomal subunit. So there's going to be an interaction here. There's going to be hydrophobic bonding. But remember, what we're trying to do here is get the protein we're producing into that membrane-bound compartment called the endoplasmic reticulum. And we've got a barrier to get across because we have that barrier of the phospholipid bilayer. So how do we manage to get, first, this protein across that barrier? And then how do we manage to get the protein incorporated into the barrier? Because we have to account for that, because we need to be dealing with transmembrane proteins. Well, the first step is to hide the hydrophobic amino acids of the signal peptide with the signal recognition particle. We'll sequester these hydrophobic amino acids from the aqueous environment by binding them to the signal recognition particle. That doesn't last very long. What will happen then is the machine of the ribosome, the mRNA, and the protein that's being produced attached to the signal recognition particle, that machine will now find its way, in ways that aren't entirely clear, find its way to the endoplasmic reticulum. Will bind to the plastic bag of the endoplasmic reticulum. So there's going to be, in the endoplasmic reticulum, two important receptors, one of which is going to be a receptor for the ribosome, and the other is going to be a receptor for the signal recognition particle. Now, at this point, the signal recognition particle bound to the protein is in the cytoplasm. When it arrives at the endoplasmic reticulum, everything's in the cytoplasm. And then there's a kind of sleight of hand between the signal recognition particle, the signal recognition particle receptor, and this gigantic protein that goes across the lipid bilayer called the protein translocator or the translocon. The sleight of hand is that there's a handoff. The hydrophobic amino acids, which had been bound to the signal recognition particle, now become bound to the transmembrane protein, the translocon. Then they get stuck there, but we start protein synthesis again. And while the amino end of this protein is stuck to the translocon, the rest of the protein slithers on by and into the lumen of the ER. And then when the rest of the protein, all the way out to the carboxy terminal, ends up in the lumen of the ER, then we cut off that part that was bound to the translocon. And now we have a free protein inside the lumen of the ER. And we'll draw how that gets done. But there has to be a mechanism here to modify this system, the simple system of threading the protein into the ER, to make a transmembrane protein that's threaded across the membrane several times. The trick here, and the part that I want you to hold on to most, is that the translocator, the translocon here, which we'll describe first as a kind of pipe through which we can stuff a thread of protein. This protein translocator is more complicated than that. It isn't just a static pipe. It has the ability to open and close, a little bit like a clamshell, or a little bit like a hairbreadth that's operated with a spring, where you squeeze the spring, the hairbreadth opens up, you let go of the spring, and it comes back together again. It's not so different from the characteristic of the translocator, where it can bind to a region of transmembrane protein, as though it were this marker. And when it opens up, then the marker leaves its binding to the protein translocator and moves out into the lipid bilayer C right next door. And we can do this for every time we run into a series of hydrophobic amino acids that are destined to become the transmembrane regions. And I'll try to explain how we come across these hydrophobic regions. So we'll have what they call start and stop sequences that will tell us to be across the lipid bilayer or not, and see if I can make some sense of that. Well, then that will bring us to the end of the idea of cells. Everything up to that point will have been a discussion of what happens in individual cell. And we could be talking about an amoeba rather than a human being. So in order to get our thoughts together about how cells work together, we have to think about tissues, because tissues are defined as a similar set of cells working in concert to perform a similar function. When we divide all of the cells in our body, we find that they can fall into 4 tissue categories: epithelia, connective tissue, muscle, and nerve. The one we're going to spend time on here, and we'll come back to over and over again, is the one known as epithelia. When you see this term epithelium or epithelia, what I want to immediately have come to mind for you is a continuous layer of cells, all standing shoulder to shoulder in a great broad sheet. It's the first idea of what an epithelium is. The cells standing shoulder to shoulder in a great broad sheet play an important role of forming an interface. They're at the borders. They are at the border between the cheek and the opening in your mouth. They are at the border between the epithelium lining your esophagus and that tube of the esophagus. There is an epithelial cell lining that's a continuous layer of cells that goes from your mouth all the way through your gastrointestinal system out to the other side, out the anus. In fact, that will be a continuous layer of epithelial cells. That's a great place to think about an interface between the outside world and your underlying tissues. We'll spend a little bit of time on connective tissue, a little bit of time on muscle and nerve, but we have several lectures dedicated to muscle and nerve, and we'll come back to those. There are some important characteristics of epithelia, though, and we have to emphasize these. First, they form the boundaries we just talked about. Secondly, they are a continuous layer of cells forming those boundaries. But more important than either of these two is they always adhere or sit on an extracellular series of proteins known as a basal lamina. The basal lamina provide a binding site for all of these epithelial cells in this great broad sheet. So they're tacked down. All of these cells are tacked down to a basal lamina. And the basal lamina is composed of extracellular proteins. This becomes important because it provides a kind of orientation for all of these epithelial cells. All of the epithelial cells in the sheet now have an orientation. They have a region that points away from the basal lamina and a region that's attached to the basal lamina. There's an apical region and a basilar region. So that orientation is a part of the language that we'll use in here, but it's important to the cells too, because those cells need to know which way to direct their proteins as they're produced, toward the apical side or the basilar side. Finally, we'll find that one of the hallmarks of all epithelia is the cells divide and make copies of themselves. They reproduce. And if you think about the lining of your mouth, the epithelium in your mouth, you know full well that if you burn the inside of your mouth, that it replaces those cells. It doesn't take very long. And the cell replication is important for all epithelia, but it also has a side effect, an unfortunate side effect, that every time a cell divides, there's the potential it becomes a cancer cell.